Please welcome Michael Blind, Ludwig-Maximilians-Universität, München.
Hello everyone, my name is Michael Blind and I'm Startup Manager in the Technology Transfer Office at the LMU Munich. Today I want to introduce our startup Tubulis, which originated from a cooperation between the LMU and the FMP in Berlin and has the mission to overcome the barriers to develop antibody-drug conjugates, ADCs, as safe and effective therapies for the treatment of cancer. Tubulis is my science startup of the year because their breakthrough technology promises to solve the over 20-year-old bottlenecks in the design, control and stability of ADCs, and equally important, their entrepreneurial success, since they were able to raise 70 million euros in their Series A and B financings in the last truly challenging 3 years. Thank you very much, and please welcome Tubulis.
Breaking the wall to cancer treatment with antibody-drug conjugates. Dominik Schumacher, Tubulis.
Every year, 10 million people are dying of cancer. And this total number has actually doubled within the last 20 years. And the number one treatment option that we have is chemotherapy. Chemotherapy where we make use of a small molecule, a potent, highly toxic small molecule, and we infuse that into the patient. And it's often very good in actually having an effect on the tumor and shrinking the tumor, but at the same time, it's always also affecting lots of healthy tissues because it's completely unspecific. And I'm sure that many of us in this room have actually experienced someone being treated with chemotherapy and seeing that these patients suffer from these strong side effects, including loss of hair, vomiting, diarrhea, and pain. And very often, actually, these side effects force the physician to stop the treatment. And that's when the cancer grows, progresses. Antibody-drug conjugates, ADCs, are a promising class of therapeutic molecules that can change this. They're composed of small, potent molecules, but this time they're firmly linked to an antibody that is targeting the tumor. So the antibody is able to find the tumor within the patient, deliver the potent molecule to the tumor gets killed, and in the best case, healthy tissues and cells remain unharmed. There is currently high momentum in the ADC space, and this is due to the fact that we actually see more and more of these molecules coming to approval. And this is great because they provide benefit for patients. But when we closely look at these molecules, we realize that they actually fall short of expectations, that they have not been able to deliver on the promise of being true targeted cancer therapies. As a matter of fact, the side effect profile, the toxicity profile of these molecules is often very similar to that we already know of chemotherapy. We are hitting the same tolerability wall in the clinics, and that actually prevents the broad applicability of this molecular class to treat patients. And the field has learned that it's actually not as expected that these molecules do not interact with healthy tissues, but they actually do interact with healthy tissues. And if we want to make a change, we need to stop these interactions. And this is exactly what we do at Tubulis. We have been developing a very broad, diverse ADC platform that enables us to design ADCs based on the requirement of the biology of the individual disease with good biophysical properties. And that enables us to increase on-target, on-tumor efficacy while minimizing healthy tissue interactions. We've been actually able to show that we can break this tolerability wall and can increase the dose by a factor of over than 5 in various preclinical models. And we are now making use of that to develop our own proprietary ADCs. And these are not projects that we have just started. It's actually, we've already scaled them to products to kilogram scale. We are ready to get these now to patients to show clinical proof of concept and hopefully show that our technologies and our products can change how we treat patients. And we're doing that together with a fantastic team. A fantastic team that is actually bringing a lot of knowledge to all the key things that we need for drug development. They've been moving from the West Coast to Munich to join our team. They've been working with large pharmaceutical companies and very relevant biotech companies, but even of more importance, these people have been developing and bringing to approval drugs that are used for everyday treating patients today. We are backed by venture capital, very experienced biotech investors. We are developing, and this is our core focus, ADCs for solid and liquid tumors. We have 30 employees, and the team is rapidly growing. And I'm super excited that we have been able to actually get access to 1,000 square meters of lab space in Munich, in the IZB. That was really challenging. And we are actually also now working with large pharmaceutical companies, a couple of them. Fortunately, I'm not able to give the names. But we are doing that because we want to bring our technology to as many patients as quickly as possible. Because we want to leverage on the therapeutic potential of ADCs and bring benefit, meaningful benefits, to cancer patients. Thank you.
Excellent timing. Okay, loads of questions. Stefan first, then Frank, please, and then Martin.
Could you elaborate a little bit more about why you have reduced the toxicity and why your payload is much better and more the right one compared to the other ADCs?
Yeah. So I think what we now have is 20 years of learning on the ADC space. And many of the ADCs has been using a similar class of payloads. And I think it's more diverse that we need to really look at what kind of payload is working in a specific indication. So we are not using just one payload, but we are using different payloads, and we're also unlocking completely new payloads. But the key question, so why can we actually reduce these interactions? There are so many mechanisms involved in driving these site-specific or target-independent interactions. And only if you are able to actually reduce all at once by having an ADC that has good biophysical properties, that actively prevents interactions with cellular membranes, then you can actually have a meaningful impact. And that's where we actually...
How do you get there? That was the question, actually. How do you get there? What's your secret sauce?
We have so we actually got inspired by nature, microtubule biology, and we are making use of a feature that actually actively, first of all, ensures water solubility of these molecules, and actively reduces interactions with target-negative cells. Because from the classical ADCs, they are very hydrophobic. And they, like a glue, they get glued to the target-negative cells, and then the cells just internalize them because they want to get rid of them, but then it's actually when we see the toxicity. And that we prevent.
Okay, thank you. Next question, Frank, please.
My question was similar, but now follow on this, it means the design of the antibody, the design of the binding, the design of the binding moiety, is that what you think differentiates you from others?
No, it's actually the product profile of the ADC in the end. You can have a very good antibody where you play with the binding properties, but then you attach a small molecule to it, and it changes the properties of that molecule. It makes it hydrophobic. It will start to aggregate. It will start to, and this causes liver toxicity, for example. And it starts to then interact with these target-negative cells. So it's actually not limited to a specific format of antibody or a specific way of binding, but it's actually very broad. And in the end, it's always our goal to have an ADC, antibody-drug conjugate, that has the same biophysical properties as the naked antibody. And that's what we aim for.
Mm-hmm. Prove this by side-to-side comparison
Many.
to already established ADC, yeah?
Many, yes.
On solid cancer.
Solid cancer, liquid cancers, various different ones, yes.
Okay, next question, Martin, please.
In terms of the cytokine release syndrome kind of things, is there any difference between your technology and the previous approaches? I mean, does that also do anything better to the patients?
So I think the cytokine release syndrome, I mean, this is a general challenge that you have when you see a very strong immune response, right? But this is not a typical challenge that we have for antibody-drug conjugates. These formats are actually highly potent, highly toxic, and you see these classical side effects like neutropenia, neuropathy, and this is really causing the limitation and reducing these, or forcing the physicians to reduce those. So this is, but something that we actually actively also work with to think new ways of how we can actually design ADCs, and then this would be more important.
Thank you. Okay, yes, no question, 12 seconds. Question, answer, thank you.
Okay, so it's here.
Yes.
How about acquire chemoresistance? It's your
Very
approach is
important.
better.
Yeah. No, that is actually very important.
Yes.
And
yes.
Yes.
Yes. Thank you very much. Sorry.